Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokade from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibault Van Zele from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN Topical Research Symposium.
Welcome Associate Professor Sarah Vreugde from Adelaide. So Adelaide, as you all know, is always been at the forefront of rhinology, clinically and in research. So Sarah has joined Adelaide. So she is a grant-funded researcher, and her main interests are diagnosing and then finding the prognostic factors and leading to the various treatment for chronic rhinosinusitis. And Sarah, is a really special thanks for your commitment today, because it's the 3rd of December for you, and it's nearly 1:15 a.m. and you're up at that time and making a presentation. So we're really, really thankful. Over to you, Sarah.
Thank you. Can you hear me? Can you see my screen as well?
Yes, we can. Thank you.
I'm actually in Belgium at the moment, so it's not that difficult for me to to give the presentation.
Oh, okay.
But
that's your timeline in Adelaide.
Yeah, so I will speak to you a little bit about our latest research on Staphylococcus aureus and their biofilms, and how Staph biofilms might be involved in chronic rhinosinusitis. And so this work was done by one of my PhD students, Gohar Shagayeg, who is now staying on to do a PhD and a postdoc in our team. So Staph aureus is it was recently published in the Lancet, actually 2 weeks ago, that Staph aureus is the most deadly bacterial pathogen, with 1.1 million deaths that happened due to Staph aureus, mainly drug-resistant Staph aureus, of course, in 2019. Of course, these kind of infections are not exactly due to the Staph aureus that might have a role in chronic rhinosinusitis. However, around 30% of the population, of human populations, are known to be permanent carriers of Staph aureus in their nasal cavity, and another 30% of human populations are intermittent carriers of Staph aureus in their nasal cavity, and it's known that those that are carriers have actually a higher risk to develop these kind of deadly infections once they enter hospitals for different reasons. Staph aureus encodes for various virulence factors, and those are often encoded by so-called mobile genetic elements like plasmids and like prophages. And Staph aureus is very well known for its potential to evade the human immune system. And Staph aureus is also known to form biofilms. So what is a biofilm? It's actually a community of bacteria, and bacteria in general in nature, they prefer to live in a biofilm rather than alone because they can work together, they can communicate better with each other, and they secrete all kinds of slimy factors that are called EPS, an EPS matrix, and that's full of proteins and polysaccharides and also extracellular DNA. And this helps them stick together, but also stick onto a surface. Within that community of bacteria, there are typically cells that are more active, that are metabolically more active, and others that are less active. And you also have persister cells. Those are metabolically not active at all. And together with the presence of this kind of slimy matrix, makes bacteria that are living in a biofilm much more resistant to antibiotics than their planktonic counterparts. And so how it works is that free floating cells, they bind to this kind of surface, which can be a biotic surface, or it can be an abiotic surface like a plastic dish in cell culture, and they then proliferate, form monolayers, microcolonies, and eventually this mature biofilm. And then when there are favorable conditions, there might be some cells that are released from those kind of mature biofilms that then can go on and stick to new surfaces, thereby expanding that biofilm. So that's how biofilm formation is occurring. Now, the problem that we have in chronic rhinosinusitis is that Staph aureus and Staph aureus biofilms are actually found in the nasal cavity of patients with and without CRS. And also, it's known that all Staph aureus can form biofilms in vitro. So the question then is, are Staph aureus biofilms actually important in chronic rhinosinusitis? So the hypothesis that we wanted to test in our lab was that not all Staph aureus are actually the same, but that there is actually strain-dependent variation in Staph aureus biofilm properties that can maybe be related to CRS disease severity and inflammation. So the aim of this study was to characterize biofilm properties of in vitro grown Staph aureus biofilms, their virulence factors, and we wanted to look at these properties in relation to immune B cell responses of CRS patients, and again in relation to CRS disease severity. The objectives the first objective was to quantify the frequency of B cell subsets in nasal polyp and mucosal samples of CRS patients and control patients using FACS analysis. So at the time of sinus surgery, we harvested polyp tissue and fresh mucosal tissue, worked them into single cell suspensions, and then did flow cytometry to characterize the B cell compartment in those patient samples. From the same patients, we then also isolated Staph aureus, and we grew those Staph aureus in biofilms for 48 hours, and then we defined their properties. So the properties meaning metabolic activity, so how active are those biofilms? What is their biomass? So how thick are the biofilms? How many bacteria are actually present within those biofilms, so colony forming units, and also how many proteins do they produce, so exoprotein production. We also extracted the DNA of those Staph aureus and then sequenced them using both long read and short read sequencing, followed by data analysis. We then looked at correlations between the different B cell subsets, CRS in vitro grown biofilm properties, and virulence genes. Can you still hear me? Yeah.
Yes, we can hear you.
Okay.
Thank you. Yeah,
Okay, sorry.
it's great.
So in terms of results, we analyzed a total of 78 patients, and we saw, as can be expected, an increase in CD3 positive T cells as well as CD19 positive B cells in CRS with nasal polyps compared to controls. This is not unexpected. We are dealing with a severe chronic inflammation. We looked at different B cell frequencies, and there was a significant increase in mature naive B cells, and this complete collapse of regulatory B cells in CRS with nasal polyps compared to controls, and also a reduction in primary B cell, memory B cell frequencies in CRS with nasal polyps compared to controls. Looking at memory B cell frequencies, again, we saw these switched memory B cells in CRS with nasal polyps compared to controls, a highly significant increase in these kind of cells. And other cell types were not so differentially present in CRS with nasal polyps compared to controls, with a slight reduction in double negative memory B cells in CRS with nasal polyps compared to controls. We also found an increase in antibody secreting cells, so that's plasma cells and plasmablasts in CRS with nasal polyps compared to controls. Now, we then looked at or measured the disease severity scores of all of these patients, namely SNOT-22 scores, Lund-Mackay and Lund-Kennedy scores. And I'm just showing you some of the results because, of course, we have results for all of these cell types in relation to these kind of disease severity scores. But to keep it more simple, I'm just showing you the results of 3 cell types, namely class-switched memory B cells, antibody-secreting cells, and regulatory B cells. And as you can see, there is this highly significant positive correlations between disease severity scores and these kind of class-switch memory B cells. The same for antibody-secreting cells, so more antibody-secreting cells, more disease severity, and more regulatory B cells, so an inverse correlation actually between regulatory B cells and CRS disease severity scores. We then looked at the Staph aureus from these same patients. So we isolated Staph aureus from these same patients, and around 40% of the patients cultured Staph aureus. So, and that was quite similar between controls and CRS without nasal polyps and CRS with nasal polyps. A little bit more in CRS with, a little bit more patients in the CRS with nasal polyp groups, but not really very different between the patient cohorts. What was different, however, was that we measured the metabolic activity of Staph aureus strains that came from these different patients, and we saw that the metabolic activity of bacterial isolates that come from patients with CRS with nasal polyps were significantly higher than metabolic activity of patients of Staph aureus strains that come from control patients. And the same is with the biomass, colony-forming units, as well as exoprotein production. So actually, Staph aureus in a CRS with nasal polyp patient has strain-dependent differences compared to Staph aureus that comes from a control patient, and those differences are only seen in biofilms. We also looked at Staph aureus biofilm properties and how they relate to B cell subsets. And again, I show you results only for some of these cell types because it's just too much information. And you can see that the normalized metabolic activity, for example, in these 2 cases, they are positively correlated and significantly correlated with the number of or the frequency of antibody secreting cells and of switched memory B cells as well. And in this case, a normalized biomass is also positively correlated with the frequency of antibody secreting cells. So if your patient is infected with a strain that has that produces thick biofilms, a big biomass, this patient is likely to have more antibody secreting cells as well. And again, we see this inverse correlation between all of these kind of metabolic activity, normalized biomass and exoprotein production with the frequency of regulatory B cells, and these are all significant negative correlations. We then also looked at how Staph aureus biofilm properties correlate with CRS disease severity. And let's just look at this first one, because there is just too much. Again, there's a lot of information on this slide. You see here normalized metabolic activity for each of the, so each data point here is one Staph aureus isolate that corresponds to one patient. So you have the severity scores for that patient, and you have the metabolic activity of the isolate, of the corresponding isolate, and you can see that there is significant positive correlations between SNOT-22 scores and Lund-Mackay and Lund-Kennedy scores and normalized metabolic activity of the Staph aureus strains that come from that patient. And the same is true for the normalized biomass, CFU counts, and exoprotein production as well. We then divided the Staph aureus strains into two groups, those that have the highest biofilm properties and those that have the lowest biofilm properties, and the properties being metabolic activity, biomass, CFU counts, and exoprotein production. And it's immediately obvious that in the isolates with the highest biofilm properties, you have all these green area here. So these are all the virulence genes that are present in those isolates, and these are the names of the isolates here in group 1 with the highest biofilm properties, and this with the lowest biofilm properties. You can see that isolates with high biofilm properties, they just carry much more virulence genes, because green is present and red is absent. We then isolated the exoproteins from the Staph aureus strains. We grew the Staph aureus strains in vitro biofilms, and we extracted the exoproteins from those biofilms, and we put that on air liquid interface cultures from primary nasal epithelial cells to look at the effect on the barrier, measuring transepithelial electrical resistance and permeability, as well as toxicity, measuring viability. And what you can see here is that result for the transepithelial electrical resistance is that from the 5 isolates in group 1, so the severe isolates, 3 of them have a significant effect on reducing the transepithelial electrical resistance, while all the isolates in group 2, so the non-severe, the isolates that come from control patients and non-severe CRS, they do not have an effect on the barrier. And the same similar result is seen with the relative paracellular permeability. So that's the permeability between the cells, where these 3 same isolates, they increase the relative permeability, while all the isolates from group 2 don't have this effect. And again, we see a reduction in cell viability of the same isolate, so indicating cell toxicity of these same isolates when exoproteins from those isolates are applied to primary nasal epithelial cells. We then look to see at the effect of in vivo, so using a C. elegans worm model. So C. elegans, it's a nice in vivo model because you don't need ethics approval to work with this in vivo model. And C. elegans, they will naturally eat the bacteria. If that bacteria is a very toxic one, the worm will die. If it's a non-virulent one, then the worm will be happy and getting really quite fat. So what you can see here is that after 24 hours, worms that ate the virulent strains from group 1, they had a significant reduction in survival compared to worms that ate the bacteria from group 2, so the non-virulent strains. And this effect was even more obvious after 48 hours. In a last experiment for this, we measured MBEC values. So MBEC values indicate sensitivity to antibiotics. We measured of the biofilms. So we measured the MBEC values for 5 different antibiotics. I just show you results for 2 of those antibiotics, augmentin and clarithromycin. So the first thing that is obvious from this is that, so here you see the different concentrations from 0, the control, to 512 micrograms per ml, which is totally not physiological, of course. And you see the fluorescence intensity indicating viability of your biofilm. So here you have a complete maximum viability, and this is the minimum viability. So what is obvious here is that even at these very high concentrations of antibiotics, you don't get a complete eradication of your biofilm. The other thing that is obvious is that in more physiological concentrations of antibiotics, you see an immense variability in sensitivity to that antibiotic between different isolates. And when you now look at how that relates to metabolic activity, for example, you see that when the biofilm has a higher metabolic activity, the MBEC values for augmentin will be significantly higher. And the same is for biomass and for colony-forming units and for exoprotein concentration as well. So in conclusion, we isolated sinonasal polyp and tissue from CRS patients and controls, and we isolated Staph aureus from those same patients. We determined the B-cell immune phenotype from those patients, and we found an increase in class-switch memory B cells and in antibody-secreting cells, and a reduction in regulatory B cells in these patients. And this related to changes in CRS disease severity scores. We grew Staph aureus in biofilms, and we could see greater biofilm properties in CRS patients with nasal polyps, as well as less sensitivity to antibiotics or higher MBEC scores. That was related to CRS disease severity scores as well. We did whole genome sequencing of these Staph aureus strains, showing that specific virulence genes were enriched in isolates that grow very thick biofilms. And this was again related to these changes in B-cell immune phenotype. So I would like to thank my group. I'm standing here in the middle, and next to me is Professor Wormold and Professor Zaltis. And the girl that did all of this, most of these studies is here, Gohar. And I would like to thank you very much for your attention.
Thank you, Sarah. That was an excellent presentation and really give a very good overview about the role of Staph aureus in chronic rhinosinusitis. And always take a pride in strong links with our department with Adelaide. Professor P.J. Vermal regularly visits our department to run the endoscopic sinus surgery and the Skull-Vest course. So it's great to see the research wing of the department getting involved with the GRN. So over to you, Thibault, if you have any questions for Sarah.
Thank you, Sarah, for this excellent presentation. I was wondering whether you have tried to eradicate these biofilms with bacteriophages. I know that you are running some studies or trials with it.
Yeah, so we are indeed using bacteriophage as a treatment, and we are going to do a double-blinded randomized control trial probably starting next year, looking to see how this is actually, looking to see how long bacteriophage treatments are necessary before you can see eradication, and whether there is an eradication or just a reduction of the biofilm and of the infection in general. So yeah, we are very much into bacteriophage treatments. Yeah. And we use nebulizers to deliver the bacteriophage, because it's a problem to deliver bacteriophage in the sinuses or any kind of medication, because nasal sprays are not ideal, and nebulizers, they often destroy the phages. So then you have to really find a system that will put them in the right place, because they are not mobile. Are bacteriophages. So yeah. So yeah, that's something that I will be able to talk to you about in about 2 years' time. I think we should have completed that trial. Yeah.
Okay, thank you. And do you think, so you showed it that it's the biofilm plays a role in a type 2 disease. Do you think it's also playing a role in type 1 disease or non-type 2 disease?
So according to what we are finding, is that if you're infected with the Staph aureus strain, and I'm only talking about Staph aureus positive patients, because we have lots of patients that are not Staph aureus positive, and we are still looking to see how the biofilm properties of the bacteria that are in that niche are actually relating to type 1 and type 2 inflammation. But what we see is that if your patient is infected with a strain that produces these kind of very virulent biofilms, then you're going to have a type 2 inflammation. I didn't show you the results of our eosinophilic panel and so on, but that's what we are finding. If you're infected with a strain that is producing very thin biofilms and that don't produce a lot of proteins, you're going to have a type 1 inflammation. And I don't say that Staph aureus is the only one that is driving the disease here. What I'm saying is that this is associating with the disease, and it might be that it's a marker of type 2 or type 1 inflammation. And we are still conducting lots of research to understand whether Staph aureus is actually responsible for driving that inflammation. That's still a question that I'm not entirely sure about. Yeah.
Okay, thank you. Interesting.
Thank you.
Are there any other questions also from the speakers or the faculty who are online?
Sarah, this is Noam Cohen at Penn. I just had a quick technical question for you. When you did your permeability experiments, when you're looking at tear, and then the next one you showed was permeability, what size, I assume you're using like a dextran to see if things are floating up or down. What was the size of the exclusion? How big of a hole was the toxins creating in the paracellular transport?
So the dextrans is 4 kilodalton.
Oh, so it's pretty small. It's very small.
Very small.
Okay.
Yeah.
Got it.
Yeah. We also have 70 kilodalton, but the cells don't like them. So we work usually with 4 kilodalton fluorescentic labeled dextrans. Yeah.
Okay, thanks.